Hello everyone, my name is Ryan Karch, and today I'm going to be talking about my summer research project in the Whelan lab through the SURE program, where we investigated electrical stimulation of the A13 nucleus as a novel therapeutic approach for Parkinson's disease. So, what is Parkinson's disease? It's a progressive neurodegenerative disorder that kills off dopaminergic neurons in the substantia nigra pars compacta. Parkinson's is characterized by motor symptoms such as rigidity, resting tremor, and freezing of gait. This video here shows a great example of freezing gait, as he's fine walking in a straight line. However, once he reaches the navigation or turning period, his gait freezes. Traditional treatments such as dopamine replacement therapy lose effectiveness over time and don't address this freezing of gait. Another method of treating Parkinson's disease is deep brain stimulation. Brain stimulation is an FDA-approved method of stimulating a deep brain structure that functions similarly to a traditional pacemaker. Traditional stimulation targets can be effective for some patients, but like dopamine replacement therapy, they still don't address the freezing of gait, which creates a demand for a new stimulation target. This brings us to the A13 nucleus, a subthalamic region within the medial zona incerta, shown in red here on the left and in purple here on the right. This region projects to locomotor regions and shows great preservation in Parkinson's patients. Previous research by the Whelan lab has shown that electrical stimulation of this region can produce a robust locomotor response in healthy rats, and that optogenetic stimulation of the A13 in Parkinsonian mice models can restore locomotor function. This leaves the A13 as a very promising stimulation target for Parkinson's patients. In our study, the primary objective is to assess the efficacy of A13 stimulation in alleviating the motor symptoms of Parkinson's disease. We hypothesize that stimulation of this region will produce a pro-locomotor response in both healthy and Parkinsonian rats. To test this, 10 rats underwent stereotaxic surgery implanting a custom-made stainless steel electrode into the A13 region. 6 of these rats received a unilateral 6-hydroxydopamine injection into the medial forebrain bundle, which models Parkinson's disease by destroying dopaminergic neurons found within the substantia nigra pars compacta. The remaining 4 rats received vehicle injections and served as our healthy controls. 3 weeks following surgery, all rats underwent parameter testing to optimize the amplitude of stimulation they will receive during behavioral testing. Following this, rats underwent a battery of behavioral testing, including tests such as the open field test, rotorod test, four-limbed stepping test, and many more. Today, we are going to look at some of the findings from the open field test, which is essentially this box here where they'll run around for about 30 minutes and we can look at their locomotor metrics. So, as expected, the 6-OHDA lesion produced a strong behavioral signature. The hallmark symptom of unilateral Parkinson's models is ipsilateral turning, or turning towards the lesion. Our lesion was placed on the right side, so we should expect turning to the right or clockwise direction, which we can see here in this video. The graph on the right shows the number of turns in control/vehicle animals and in our 6-OHDA or Parkinson's group. As you can see, Parkinsonian rats underwent significantly more ipsilateral turns than contralateral turns, which was not seen in our controls. This shows that the lesion in the substantia nigra pars compacta had its intended effect. Moving on to the stimulated open field test, in healthy controls, stimulation did modulate locomotion, but not in the way we expected. The left graph here shows average speed across 3 1-minute stimulation periods. 1 minute pre-stimulation, 1 minute of stimulation, and then 1 minute post-stimulation. The middle graph shows the locomotion duration across the same 3 time periods, and then the rightmost graph shows bouts of locomotion across those same 3 time periods. In healthy controls, all 3 of these metrics showed significant increases in the post-stimulation period. However, no changes were observed in the stimulation period, which is very puzzling. Another concern is our 6-OHDA group, which showed no significant changes across all 3 metrics. While some 6-OHDA animals showed partial recovery, most showed little to no improvements. Quick flash of the references. And unfortunately, that's all the time we have today. If you are interested in this topic, please come to my poster at the Student Union Research Symposium, where I'll be showing more behavioral results and additional post-hoc tissue analysis. Thank you all for watching. I would also like to thank the Whelan Lab for serving as a perfect introduction to the world of research, as well as the University of Calgary and Hotchkiss Brain Institute. And of course, a huge thank you to NSERC for funding me, and have a great day, everyone.